Hello everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover Laboratory 24, the ECG or electrocardiogram and pulse discussion. So let's start off with a little bit of background information. So the heart muscle acts as an integrated unit. We want our heart to beat as efficiently as possible. Our hearts start beating before we're born, and they beat for many, many years, and they don't get a break. We want to exercise our heart. We want it to work hard sometimes, but we want it to work hard for 1 to 2 hours, 4 to 5 times a week, not every moment of every day. So we need our heart. We need the muscles of our heart to work together. We need them to contract in a coordinated manner. The heart has a functional syncytium. It's a network of interconnected cells. When one heart muscle contracts, it passes that connection to the adjacent cells via direct cytoplasmic connections. Intercalated discs contain gap junctions. Gap junctions are those direct cytoplasmic connections, and the intercalated discs are in between each of those cells, so these parts. Signals pass directly, and it helps coordinate contraction and relaxation. Autorhythmic cells. So all of our cells, all the cells in our heart can actually depolarize just by themselves without outside signals. But there's a group of cells that does it the fastest called the SA nodes. And the SA nodes, when they depolarize, they pass that, when they contract, they pass that onto the adjacent cells, which pass that onto the adjacent cells, which passes that onto the adjacent cells through those intercalated discs. So those are the autorhythmic. They can set their own rhythm. They generate their own action potentials. There's a few different nodal tissues, which are the areas where that depolarization happens the fastest: the SA node, the AV node, the V node. Whichever one's the fastest will kind of reset the other ones as that depolarization passes through those cells. So as long as the SA node's working correctly, the SA node is that fastest pacemaker that sets the rate. Perfect. Here's the SA node, the AV node, the ventricular node. So the SA node, the contraction will start here, spread throughout the atria. Perfect. So start here, it'll spread throughout the atria, causing the atria to push the blood into the ventricles to completely fill them. It's going to eventually get to, remember, the interatrial septa allows for that depolarization to pass through. The atrioventricular septa does not allow for that signal to pass through. So the depolarization event is going to reach the AV node, where the conduction is going to slow a little bit. Then it's going to be passed to the bundle of His, to the left and right bundle branches, to the apex, which is this part of the heart, and then it's going to spread out among the Purkinje fibers, where it starts to contract the heart from the apex, kind of going up, and we get ventricular contraction. The ECG provides an electrical view of the heart. Sometimes it's referred to as an EKG, ECG or EKG. It's an electrical recording of the heart. We can put sensors, we can put electrical sensors on our extremities, and we can actually detect the electrical activity within our heart via those sensors. Our blood has a lot of electrolytes in it, so it carries electricity well. This is called volume conductance. Volume conductance is why we're able to sense cardiac action on our extremities. Electrocardiograms are very diagnostic of a number of things. They can give us a lot of information about the function of the heart. They can tell us heart rate, which we'll be doing in this lab. They can tell us about previous myocardial infarctions, telling us about conduction abnormalities and the size of different heart chambers. The ECG consists of a series of waves, segments, and intervals. So here's some vocab words that you should be familiar with. A wave is a deflection above or below baseline. So right here we have the P wave, it's a deflection above the baseline. Right here we have a T wave, deflection above the baseline. A segment is the baseline between waves. And an interval is a combination of waves and segments. So the PR interval, combination of waves and segments. The QT interval, combination of waves and segments. The electrocardiogram activity corresponds to events in the cardiac cycle. The P wave is where we get atrial depolarization. That's where we get atrial contraction. That's where we get atrial systole starts at the P wave. The QRS complex, two things happen. I want you to pay attention to this. Students, this is a commonly missed thing on quizzes and exams. Two things happen in the QRS complex. Atrial repolarization, so atrial relaxation, so the atria contracts here, atrial systole starts here, and it ends here in the QRS complex, so atrial repolarization or atrial relaxation. When the atria relaxes, we also get ventricular depolarization. Ventricular depolarization or ventricular systole or ventricular contraction. That ends at the T wave. T wave is when ventricular repolarization ends. So you can see right here, muscle action potential for the atria ends right when the ventricular muscle action potential begins. Perfect. Right here you can see the corresponding parts of the ECG along with how that depolarization event is passing through the heart. ECG. So to determine heart rate in the ECG, we need to determine how long it takes for one cardiac cycle. How long does it take for one cardiac cycle? So we pick one part of the cycle and we follow it all, and we measure the time it takes to go all the way through the cycle to the next. And the part that we actually use is is R. So we do the R to R interval. So we start with R right here, and we measure all the way to the next R. R is really obvious, so it's a good spot to choose to do the R to R interval. So heart rate, one cycle is measuring R to R. A normal resting heart rate is 60 to 100 beats per minute. Bradycardia, that's a vocab word. Anything less than 60 beats per minute is considered bradycardia. Sometimes bradycardia is a normal thing, it's fine. And sometimes bradycardia is not a good thing. If bradycardia is because of just how that person was built genetically, they just have a slow SA node or very active parasympathetic nervous system, it's fine. If it's because they exercise and they're able to pump a lot of blood out of their heart per beat, they have a very strong heart, they have a large stroke volume, that's fine also. If it's because of a disease state, not the greatest thing. Tachycardia, beats of 100 or more per minute, could be genetic, fast SA node, or it could be disease. Generally, it's not a good thing. Just if at rest, you have 100 beats per minute. So sinus rhythm is actually the name given to just the normal rhythm. So the SA node rhythm. When the SA node's in charge and we just get normal conduction, sinus rhythm. Perfect. The normal pathway. Sometimes the SA node is not in control. Sometimes there's conduction abnormalities, and the SA node doesn't start the depolarization event, and that depolarization event doesn't travel smoothly through the rest of the heart in the pattern it's supposed to. Remember, there's other nodes, and really all parts of the heart could spontaneously depolarize. Sometimes there's an issue where a different part of the heart starts a contraction. It's an arrhythmia. It's a faulty node or conductance. Ectopic means out of place. So like an ectopic pregnancy is when a baby starts growing in the wrong place. An ectopic or out of place pacemaker. So this is what I was talking about when a different part of the heart could start a depolarization event, could start a contraction event. It's called fibrillation. When there's an ectopic or out of place pacemaker, so some other part of the heart is starting the contraction cycle. Fibrillation. It's going to cause the ECG to look very different. A heart block is when there's a conduction problem. So sometimes the atria and the ventricles contract independently, that the normal depolarization event isn't able to go to the AV node and pass through the bundle of His and pass through the left and right bundle branches. There's some sort of conduction problem with the passage of that electrical event, and it's called a heart block. Sometimes there's actually a premature ventricular contraction, or PVC, and that's when the ventricle contracts before it's completely full. So while the atria is still contracting and still pushing blood into the ventricle is still filling, but it's not full. The ventricle is not full of blood and it contracts. This is going to mean that you're going to take more heartbeats to push the same amount of blood. You want to pump as much blood per heartbeat, so it requires the fewest number of heartbeats. This is going to make your heart work harder than it needs to. It's going to decrease efficiency, and it's not going to be good long term. So filling and pumping is uncoordinated. It reduces cardiac output. So right here, you can see atrial fibrillation. It's going to change how the ECG looks. Now, I'm not going to give you a picture of an abnormal ECG and ask you what's going on. I might give you a photo of a normal ECG and ask you what's going on at each place. In fact, you should know. That's actually a good way to practice. Just look at a normal ECG picture and tell me what's going on at each place. That's a great way to practice for lab and lecture. So, point of this slide though is you can tell a lot by what the ECG looks like. It's very diagnostic. So now let's get back really into what we were doing with lab. So we were seeing that the ECG changes in lesson 5 while sitting compared to taking 3 deep, slow breaths compared to exercise. So before we jump into this, how do you think the ECG is going to change with sitting? How do you think it's going to change with 3 deep, slow breaths? And how do you think it's going to change with exercise? What parts are going to change specifically? What parts of the ECG? Is there going to be any change at all? All right. So here are the numbers with sitting. You don't need to memorize the sitting numbers. Know what part is represented with the wave though. You know, what happens at P wave, what happens at QRS. Perfect. All right. So let's look at the heart rate for each of these parts. So in sitting and resting, 74 beats per minute. With a deep inhale, it goes up. With a deep, and then as we exhale, it goes down. Why might heart rate go up during the inhalation and go down during exhale? Why might heart rate change while we're breathing? So our heart is very responsive to the demands of our body. It's very responsive. And inspiration is an active process. We actually use a lot of energy to take a deep breath. It's an active process. It uses muscles. When we use muscles, we need to use ATP. When we use ATP, we need to produce more ATP. The production of ATP, as we learned for section 2 of this class, it produces carbon dioxide, and carbon dioxide is the strongest driver for the desire to breathe in this class, but we're talking about heart rate. When we produce more CO2, we need to pump our blood faster to get new nutrients to those places that need it and to get the waste products away. And our heart is very responsive to that. And it will actually increase heart rate when we take a deep breath. It'll actually go back down when we exhale. Occurs during normal breathing, a change of about 10 to 20 beats per minute. A change of about 10 to 20 beats per minute will happen just during normal breathing. We breathe in, the heart goes a little bit faster. We exhale, the heart goes a little bit slower. It's called the respiratory sinus arrhythmia, RSA, respiratory sinus arrhythmia, and it happens normally. Remember, inspiration is an active process. We need more nutrients to those areas. We need to get waste products away, faster heart rate, expiration, passive process. Now with exercise, I think most students get this part. With exercise, we're using a lot of muscles. We're making a lot of ATP. We need nutrients in our muscles. We need to get waste products away. Exercise increases heart rate. But what part of the ECG, what aspects change to produce this exercise? Does everything happen faster? How does the ECG change? Does everything happen faster? Does one part happen faster? Does the amplitude change of any parts? Well, let's take a look at this. So we've got the QT segment, which is ventricular systole. We're going to go from the Q to T. The ventricle contracts in the QRS complex. The ventricle relaxes in the T wave. So this is ventricular systole. In lab, we're going to measure this time. How long does ventricular systole happen? And then we're going to measure the TQ interval. So going from ventricular relaxation to the end of ventricular relaxation, how long does ventricular diastole happen? So QT is ventricular systole, TQ is ventricular diastole. And what we're actually going to see is that under normal resting circumstances, the QT interval, or ventricular systole, is about 1/3 of the heart cycle. And the TQ, ventricular diastole, is about 2/3 of the heart cycle. When we exercise, the QT, ventricular systole, is not going to change much. It's not going to change much. So ventricular systole, very slight change between ventricular systole time when sitting and ventricular systole time when exercising. Our heart wants to beat as efficiently as possible every single time. It's going to beat as efficiently as possible whether it's sitting or whether you're exercising. The part that changes is diastole, the time in between beats, this rest period. That's what changes. The time between beats goes down a lot. So 0.54 seconds to 0.24 seconds. Ventricular diastole changes a lot. Ventricular systole pretty much doesn't, or very, very slightly changes. So when we calculate MAP, remember that how we calculated MAP for previous lessons, previous lectures? We said 1/3 systole plus 2/3 diastole will give us our MAP, our average mean arterial blood pressure. We said 1/3 systole, 2/3 diastole. That's because of this right here. Because at rest, 2/3 of the time we're in diastole and 1/3 of the time we're at systole. When we exercise, the equation to calculate MAP actually changes to 1/2 systole and 1/2 diastole. Because diastole, when we exercise, is only about half of the cardiac cycle, and systole is about half the cardiac cycle. So, all right, now let's look at pulse. So, the thing we use to calculate pulse is a plethysmograph. It's tough to say. Plethysmograph. I'm probably saying that totally wrong. It measures the pulse pressure. When you feel your pulse in your neck, you feel that expansion of the artery. When you feel your radial pulse in your wrist, you feel that expansion of the artery. That's what this is measuring. It's not measuring the electrical activity of your heart. It's measuring that expansion of the artery that it feels in the finger. So we're going to measure ECG and pulse while sitting with our hand on the table. We're going to drop our arm below the waist and then hold the hand above the student's head. How is the ECG going to change in these different scenarios? How is pulse going to change in these different scenarios? Another thing we can think about is how are they going to compare? Is heart rate, according to ECG, going to be different or the same as our pulse rate? Our pulse rate is that feeling of pressure distally in our arteries. The ECG is going to be the actual electrical contraction of our heart. Are they going to happen at the same time? Are they not? Are they going to happen at the same rate or not? All right, we'll start with heart rate and pulse rate. So we measure the R to R interval. So we measure one cycle, and we measure the pulse interval. So the amount of time from one pulse wave to the next pulse wave. We get 0.78 and 0.77. So basically the same. And then we extrapolate that out to a heart rate and a pulse rate over 1 minute. So we get 77 beats per minute heart rate, 77.9 beats per minute pulse rate. It's the same. You know, any difference is just error in measuring. When you have a contraction of the heart, that's going to produce that pulse pressure wave that moves through our arterial tree that is felt as the pulse in your finger. So there is one contraction of the heart, there is one pulse pressure wave. So it'll be a 1-to-1 ratio. Now, are they going to happen at the exact same time? All right, so let's move on to the next slide. So when we move our arm, when we hold our arm below our waist, or when we hold our hand above our head, how is that going to affect the electrical activity of our heart, and how is that going to affect pulse pressure? Well, the ECG is actually unaffected by position. I mean, maybe your heart rate goes up a little bit because you're using energy to hold your hand up above your head. But the amplitude, the height of the ECG, the height of each wave in the QRS complex is going to be unaffected. Heart rate is going to be basically unaffected, and any change is just going to be because of the energy you use holding your hand above your head. But amplitude is going to be unaffected. Pulse is going to be altered by position. The rate, again, is only going to be different because of the energy you're using to hold your hand above. And remember, pulse rate and heart rate with ECG, it's going to be 1-to-1 ratio, basically the same thing. What's going to be different is the volume of blood, the amount of expansion that you have. The amount of expansion that you have. When you have your hand down below, resting down below, there's going to be gravity is going to pull blood downward. You're going to have a larger pulse when your hand is down by your waist. You're going to have a larger expansion, pulse pressure wave. When your hand is up above your head, your blood's got to fight against gravity. There's going to be a smaller pulse pressure wave. Smaller pulse pressure wave. So hand below the heart, QRS complex is unaffected, same amplitude. QRS amplitude, basically the same across. Any error is probably just due to measuring error. Relative pulse amplitude, 3.56 when the arm's relaxed on the table in front of you. 8.24 when your arm is down. 0.23 when it's up. It goes down a lot when it's up. And that's because when your hand is down, blood pools in your hand, you have an increased volume and increased pressure. When your hand is up above your head, gravity pulls it down out of your hand. We're going to have a smaller volume, a smaller pulse pressure. Hand is above. Perfect. Pulse is moving uphill. All right, there's one more thing I want to point out. Does the contraction of the heart happen at the same time that you experience the pulse pressure wave at the distal part of your finger? So let's look at the data. Right here is where ventricular systole begins. Right here is where ventricular systole ends. The pulse pressure wave reaches its peak right over here. No, they do not happen at the exact same time. The heart is going to, the ventricle is going to contract, and it's going to, push blood. It's going to eject blood from the left ventricle into the aorta. And that pulse pressure wave has to physically travel from your aorta to the distal part of your finger. And that travel time is going to cause the pulse pressure wave to be experienced just after when the heart beats. So there is going to be a little bit of time delay in between the end of ventricular systole and when that pulse pressure wave is experienced. All right, as always, if you have any questions, please email me. I'm here to help, and I will see you in the next video.